Hello everyone, welcome back to the Misostudy.com, and in this session we are again discussing some important question from the NEET biology. As the NEET is again after a couple of weeks, your NEET test and NEET exam is there. So for preparing we are discussing the next session, and in this session we are discussing about the cell cycle and the cell division. And in this session we are discussing the questions one by one. Let's discuss the first question from cell cycle in the cell division, that is, core histone proteins are synthesized during which of the following phase of cell cycle? Option A, S phase, B, G2 phase, C, G1 phase, and D is both A and C. When we discuss about the core histone protein, that is during G1 phase, also known as post-mitotic phase. G1 phase is also known as a post-mitotic phase, that is known for cell grows. In this phase, cells generally grows, synthesis of RNA, ribosomes, enzymes and proteins. That proteins are necessary for the DNA synthesis. That generally necessary for DNA synthesis occur in this phase. When we discuss about the S phase is also known as a synthetic phase, and in this synthetic phase there is a replication of DNA is seen. And for the replication of DNA histone proteins are required and generally synthesized also in the S phase. It's take about 30 to 50% of total cell that is synthesized in the body. The next phase and important phase of cell cycle is G2 phase. In G2 phase is also known as a pre-mitotic phase. And in this phase there is a division named as nucleolar RNA. Other than nucleolar, it generally involves the formation of ribosomal RNA. Next involve the messenger RNA, and all these are synthesized in the G phase. In the discussion we are discussing three phases. First is a G2 phase, next is a S phase, and third is a G1 phase. Along with the discussion, we discuss about the in which phase what process is going through. According to the discussion, the core histone proteins, as discussed in the discussion, histone proteins are formed and synthesized in the S phase. So according to the discussion and the question, the right answer for the question is option A, that is S phase. Core histone proteins are synthesized in the S phase of cell cycle. Let's discuss the next question, that is, cell division or mitosis is a normal process in living cell, but sudden and abnormal mitosis in an organism will frequently result in option A, zygote, cancer, new organ and syncytium. When we discuss about the cell division, there are two kinds of cell division named as mitosis or meiosis. Meiosis is known as a reductional division and mitosis is known as a equational division. In the mitosis is a normal process where single cell a single cell divides to form two daughter cells. Single cell divides to form the two daughter cells, and these divisions are because of abnormal, but in the these are the case we discuss in the normal process, but in the case of abnormal mitosis it may occur in the certain cells. It may occur in certain cells which result in causing cancer to the body. When mitosis undergo the abnormal process of certain cells in which certain cells continue to divide, and that generally proceeds to form the cancer and cause cancer in the body. So how cancer arise? Cancer arises from abnormal to the uncontrolled division. Abnormal to uncontrolled division that generally occur in the body, and due to which they destroy the surrounding tissue. They destroy the surrounding tissue. In the cancer cells generally lost the unusual control over their growth and division. So in that way, normal cell and cancerous cell are differentiate from each other. In the cancer cell, they generally lost their control and for their growth and division, and in the normal cell, they generally control their growth and divide into from one single cell divide into the two daughter cells. So according to the discussion, the right answer for the question is about the cancer. Cancer is what? Abnormal growth of the mitosis shown by the mitosis, and the cancer cells show unusual control growth over their growth and the division. So the right answer for the question is option B, that is cancer. Let's discuss the next question from cell cycle and cell division, that is spindle fiber unite with which structure chromosome? Option A, chromocenter, B, chromomere, C, kinetochore, D, centriole. When we discuss about the spindle fiber, first if spindle is microtubular apparatus that appears in many eukaryotic cells that are present at the beginning of nuclear division. And this is generally used in order for separation of chromosome. Separation of chromosome. Chromosome is being attached to the spindle fiber. Chromosomes attached to spindle fiber by the term name as a centromeres. By the term name as a centromeres. So with the help of this, two types of spindle fibers are distinguished from each other on the basis of their interpolar fiber, which generally stretch continuously from pole to pole of spindle. Till now we discuss chromosomes are attached to the spindle fiber with the help of centromeres, and two types of spindle fibers are interconnected with each other by the interpolar fibers, and these interpolar fibers are stretched continuously from pole to pole of spindle, and with the help of kinetochore fiber, which generally stretch from pole to centromere. Centromere is also known as a kinetochore. Kinetochore of an individual chromosome. The mechanism by which chromosomes moves and spindle fibers contract remains unclear. When we discuss about the cells of animals and lower plants, that generally possess the centrioles. Which act as organizer reason for spindle microtubular formation. But in that case, centrioles are absent from the cells of higher plants. So this is the discussion about the spindle formation, and according to the discussion, the right answer for the question is option C. That generally includes there are two types of fibers. First is the fibers includes centromeres, and these centromeres is also includes the specific fiber, that is kinetochore. So kinetochore is the right option in this case. So option C is the right one in this case. Let's move on and discuss to the next question from cell cycle and cell division. That is question number 4 is, which of the following structure will not be common to mitotic cell of a higher plants? Option A, cell plate, B, centromere, C, centriole, D, spindle fiber. When we discuss about the first is about the centrioles. The centrioles occur nearly in all animal cells and in motile plant cells. In motile plant cells. Motile plant cells, for example, as zoospores of algae, and next is sperm cells of fern, and third one is in the case of motile algae. All these three are known as zoospores of algae, sperm cells of fern, and motile algae are come under in the motile plant cells. They generally possess, or centrioles are absent in amoebae, prokaryotic cells, higher gymnosperms, and in the all angiosperms. Centriole are present only in all animal cell and few plant cell that generally possess the motile property, such as the zoospores, sperm cells, and motile algae. Apart from that, they are absent in all other plant kingdoms and plant phylum. In this, there is a phase known as interphase. Interphase is also known as a undividing cell. Undividing cell that generally has a pair of centrioles. A pair of centrioles is also known as a diplosome, and that centriole and diplosome is usually present near to nucleus. They lie in the small mass. Of specialized, distinctly stained cytoplasm. That cytoplasm generally lack the cell organelles. The centrioles and centrosphere. The centrioles and centrospheres are together referred as the centrosome. Centrosome is known as when centriole attach with the centrosphere. Before cell division, before cell division, the centriole centrioles duplicate so that a dividing cell has a pair of centrioles. Before cell division, cell centrioles undergo the process of duplication so that every dividing cell get a pair of centrioles, and that centriole present at each pole of spindle. So, according to the discussion, spindle fiber, cell plate and centromere. Spindle fiber, cell plate and centromere are present in all plant cells. So according to the discussion, the right answer for the question is centrioles. In centrioles, along attached with the centrosphere, they form the centrosome, and the spindle fibers, cell plate and the centromere are present in all plant cells, but centromere is only present in motile plant cells and absent in amoebae, prokaryotic cells, gymnosperms and all angiosperms. Angiosperm is known as a flowering plants. So according to the discussion, the right answer for the question is option C, known as a centriole. Let's discuss the next question from cell cycle and cell division is the characteristic of meiosis is that it, option A, can produce 4 cells having chromosomal number equal to mother cell. B, occur in all types of cell. C, maintains the constant number of chromosome in a particular species. D, promotes growth of animal body. As we discuss, in every body there is a two kind of a division, meiosis and mitotic. Meiosis is a divisions or a type of cell division, by which germ cell named as eggs and sperm are produced. So meiosis is the basic division and it is also known as a reductional division. And it is a type of cell division due to which germ cells are produced, for example sperm and egg. Meiosis involved two successive nuclear divisions and one cytoplasmic divisions, and that generally produce the four haploid cells. The first division in meiosis 1 is the reductional division, and second division in the meiosis 2 is just like the mitosis and it's also known as that generally separates the chromatids that chromatids which are produced at the division 1. During the division 1, there is a meiosis is further categorized into two types. In first division occur at meiosis 1 is also known as a reductional division, and second division occur in meiosis 2 that generally helps to separate the chromatids that are produced during the meiosis 1. Meiosis reduce the chromosome number in half. Chromosome number in half during the formation of gametes. During the formation of gametes. That's why the division is also known as a reductional division, because they generally reduce the chromosome number to the half. That's why it is also known as a reductional division. During the first division, that is the reductional division, the chromosome pairs are partitioned so as each gamete or spore contains one of each chromosomal pair. That is, the condition is haploid. First division is also known as a reductional division, in which chromosome pairs are partitioned so that each gamete contains one of each chromosome pair. That is, the condition is haploid. And when haploid gametes unite during fertilization, the resulting zygote is facing the condition that is diploid. First, they convert the chromosome number to the half and the condition is haploid. Then the two haploid gametes unite during the fertilization and the zygote condition is diploid. Hence, they receive one chromosome of each pair from each parent. Since they generally received one chromosome of each pair from each parent. So according to the discussion, the right answer for the question is about the meiosis. So this is all about the discussion. Meiosis is also known as a reductional division and mitosis is known as an equational division, because in meiosis the first they reduced their number of chromosome and turned into the haploid condition, and when two haploid gametes unite, the zygote is facing diploid. So according to the discussion, the right answer for the question is maintain the constant chromosome number in a particular species, because it contains two divisions, meiosis 1 and meiosis 2. Meiosis 1 is also known as a reductional division, and in meiosis 2 is also known as a equational division, and hence also work as a separates the chromatid that is produced at the meiosis 1. So option C is the right one in this case. Let's discuss the next question from cell cycle and division, that is Lampbrush chromosome are seen in which typical stage? Option A, mitotic anaphase, B, mitotic prophase, C, mitotic metaphase, and D, meiotic prophase. When we discuss about Lampbrush chromosome that generally occur in prophase of meiosis 1, that is also known as a meiosis 1 is also known as a reductional division, and Lampbrush is also known as a highly elongated special kind of synapsed mid-prophase and diplotin chromosome, also known as a diplotin chromosome. Lampbrush chromosome is generally present at prophase of meiosis 1, and they are also known as a highly elongated special kind of synapsed mid-prophase diplotin chromosome that generally undergo the process of crossing over. Lampbrush chromosome always occur in pairs. Each chromosome of a pair has double main axis, and that double main axis due to presence of two elongated chromatids. Lampbrush is always present in a pairs, and that pairs are attached to the double main axis, and that double main axis are work due to presence of two elongated chromatids. Both the chromatids bear rows of large number of chromosomes. They generally bear large number of chromosome. When we discuss about two adjacent chromomeres, that generally give the lateral projections. Or appear like the loops. The lateral loops provide a test tube or lamp brush like Lampbrush like appearance of chromosome, that's why the name of chromosome is Lampbrush chromosome, because in this there is an arrangement of two adjacent chromomeres in the lateral projections or the loops. These lateral loops are provide a test tube or lampbrush like appearance, and after that the name of the chromosome is Lampbrush chromosome. Lateral loops take part in rapid transcription of DNA to mRNA, messenger mRNA, meant they generally help in synthesis of yolk or other substances that generally required as or important for growth and development of meiosis. So lateral loops generally help in the rapid transcription of DNA to produce the mRNA strands, and that is apart for the transcription they also involved in the synthesis of yolk and other substances, and hence it help in the growth and development of the meiosis. So according to the discussion, the right answer for the question is option D, that generally includes the function of the Lampbrush chromosome, or the position, that is Lampbrush chromosome, are seen in the meiotic prophase. As we discussed in the meiosis prophase 1 is generally the position in which Lampbrush chromosome are present. And why we name it as a Lampbrush? Because they generally arranged in the lateral loops, and these loops are appear like the Lampbrush shape. Hence, after the name of its shape or appearance, the name of the chromosome is Lampbrush chromosome. So student, this is all about for the session in the cell cycle and the cell division. Hope student you like the lecture. So stay tuned with the Misostudy for such more sessions. Till then, take care and thank you very much.